All right, so welcome back. Uh, those who are joining us now. So before break, we focused on connecting AI to data, and now we are going to shift from access to analysis. And Dr. Julie Tacker from University of Rochester, she will lead us through a guided analysis of an AI-ready dataset and show how these capabilities come together in a practical research workflow. With that, I will welcome Julie and turn it over to you.
Thank you, Sanchita. I'm happy to be here. Thank you for joining this guided tutorial. So for in next 50 minutes, what I would like to do is talk about the data sets and some of the goals behind developing this data set. So I'm going to spend few minutes, maybe 10 minutes, talking about the data set using this presentation. And then we will move on to the guided tutorial in Google Colab. And we'll try to run the script together to see how we can actually use the dataset. So this immune signatures data resource, which is an AI-ready compendium of systems vaccinology datasets, was developed by myself and Joanne DeRay RC. She's assistant professor in the Department of Pediatrics at Harvard Medical School, and I'm professor at the Department of Microbiology and Immunology at the University of Rochester. So this project was developed as a part of US AI innovation objective. So we had a pilot award from NSF where this consortium or the data was developed. It's called NAIRR project or pilot award. And as part of this pilot award, there were 3 data sets that were developed, 10K immunomes, COVID-19, and today we'll be talking about immune signatures data resource, which was developed in collaboration with the Human Immunology Project Consortium. And I'm going to talk a little bit more about what this data consists of. Overall, it has integration of 30 studies and 1,405 subjects. The original paper is published in Scientific Data, and I'm also going to show you how you can access the dataset using ImmPort database. So brief introduction to the systems immunology, which is an holistic approach to understand humans' immune response to multiple different antigens or diseases or infections. The key tools that we use in systems immunology are omics data sets such as transcriptomic, proteomics, and metabolomics. And the applications are in the fields of vaccine development, disease modeling, and personalized medicines. And what we would like to do using these large-scale data sets is to predict human immune responses by analyzing complex interactions across cells and molecules. This data was generated, as I said, as part of Human Immunology Project Consortium, which is NIH, NIAID funded Systems Immunology Consortium. So this is the original study, which was the first author was Joanne. It's an immune signature data resource, has 53 cohorts, 30 studies, and 24 vaccines. As I said, it has 1,405 participants and 4,795 transcriptional profiles. The way the data set was developed was integrating information across HIPC centers and also some data from non-HIPC centers. And the gene expression omnibus was combined in ImmPort through specific study profile information and then was analyzed using a common quality control pipeline which also included batch correction and age and biological sex imputation. And at the end, this paper was published in Scientific Data. So this gives you overview of what is in the data set, right? So vaccine responses are measured by HAI titer, non-neutralizing antibodies, and ELISA. And these are the number of vaccines that we have. And for each vaccine or for multiple vaccines, we have a time series data. And on the right, you can see how the samples are distributed across multiple time points. And one thing to note is that we have a good amount of data at the baseline, so which we can use as a control to really understand the vaccine response. And we have multiple different transcriptomic data. So as you can see in the Venn diagram, the transcriptomic data was one of the highest number of assay that was run. And here is again a brief overview of what we can, the type of analysis we can do using this data set, right? So since we have multiple time point, you can start to ask, okay, what genes are induced across different vaccines, or what modules are expressed across different vaccine. So here I'm showing you each row is a gene expression module, and column is the vaccines, and you can see, okay, what modules are induced on day 1, day 3, and day 7, and so on. So with the goal of automating the workflow, we developed this AI-ready data set now, which now you can use openly and develop different types of analysis. And as I said, it was developed as part of an AI-RR pilot, which is the funding that is coming from Office of Data Science Strategy. One of the things ImmPort team did really well was to develop this assessment template that allows us to assess the AI readiness of the data set. And I'm not going to go in detail explaining this template, and you can look at the template on ImmPort database. But what this template does, it allows you to characterize your own data and its ability to be used in automated workflow. So the data is ranked based on data preparation, data quality, data documentation, and data access. To give you an example, when we started developing ISDR data set, the scores for some of these metrics were lower. And as we developed completed the data set, the scores improved over the period of 1 year. So this gives you idea about how you can use this assessment to develop AI-ready data sets. So then in this tutorial, what I want to do is I want to use a subset of samples, and we will use these samples to study how vaccine response can be predicted. So here I'm showing you that there are many participants, and some of them are 60 and above, and others are in the range of 18 years and 49 years old. So in the tutorial, we only use these young adults in this age range. And we looked at their antibody responses. And here is an example. On the right-hand side, there is an example of how antibody responses are distributed. And just to remind you that this is a time series data for which we have multiple time points. And this data was basically used to decide who is low and who is high responder. And this is a sort of data that you have in the data set. So for each participant, there is a metadata associated with the participant. So of course, there is identifier, then study success accession number, and arm accession number, and cohort assignment. Then there is demographic information such as race, ethnicity, sex, and age imputed. And then there is a vaccine metadata, such as the type of vaccine, type of adjuvant, pathogen, and the assay by which immune response after that vaccination was measured. And then at the end, the most important is how we can measure vaccine response, right? So you can do multiple different types of analysis. You can use continuous variable to define the vaccine response. But here, what we are going to use is MFC-P50, which is by using this MFC-P50, we divide low and high responder, which was defined by using quantile threshold specified by the 30th percentile cut point. Other than MFC P50, there are other measurements that are available in the data sets, which we will not be addressing in this tutorial, but some other measurements are max RBA, which is participants' immune response classification based on maximum residual baseline adjusted response. Then max strain MFC, which is viral strain that produced a maximum fold change, and max strain RBA, which is viral strain that produced the maximum baseline adjusted response. Again, this gives you overall introduction to the data set. All of this information is available in the package that you can download from ImmPort, and there is a README file that you can use to get this information. So with that, I'm going to stop, I think, the presentation. And next, I would like to show you how to access the data on ImmPort website. Okay, so I hope you can see my screen. This is the ImmPort website. If you go to the resource page, you can go to this in AIRR, AI-ready datasets. You can click on it, and then these are the 3 datasets that I mentioned in my talk. And then you can download this dataset from here. This has the data in RDS format. There are 2 RDS formats that we will use in the tutorial. There is a README file along with some other information. If you go to this link, you can see the assessment scores that we gave to this data set, the assessment that I was talking about, explaining the AI readiness of the data set. So now I'm going to move on and run the tutorial. So to do that, I think this is the folder that is shared with you, workshop materials for participants. So if you go to this folder, you can open this folder called Guided AI Ready Dataset Analysis. Let's go ahead and open it. And then this is the tutorial, Guided Vaccine Analysis tutorial. If you click the 3 dots on the right-hand side, you'll see this menu. And in the Open With drop-down menu, you can open the file in Google Colab. So I'm just going to wait a couple of minutes to see if anyone has questions. I see there are 11 chats in the chat, but I am not seeing it right now. Okay. Anyone has any questions at this point? So this is a tutorial in R. So let's go ahead and set up the runtime to R. So if you go to the runtime and click this change runtime, you can choose the runtime type to be R. Okay, with me? So earlier here, you have information about the data set that I talked about. And then we are going to try to install the packages that we need for this tutorial. So now this step takes a little bit of time. And also this is a good time to ask questions if you have any. If you cannot open the Colab the way I showed you, there is also another option of downloading and uploading, and I'm happy to address that as well.
Yeah, please feel free to raise your hand and we can unmute or post message in the chat if you're experiencing any issue opening the notebook.
So just to talk a little bit about the tools. So these are some of the packages that are mostly needed for basic statistical analysis. It has caret, random forest, proc, ggplot, dippler, forcats, and reshape2 and purrr. These packages are available as in basic R. And then there are two packages that we are using. Those are Lima and BioBase. And those are offered as a part of BioC Manager. And these are the two RDS files that I was talking about. We are going to need in this tutorial all im data with response.rds and norm iset. Norm iset has basically the gene expression data, and the metadata that I was talking about is in this other file called all mdata with response. So if you want to group the patients differently than what I'm doing in this tutorial, you can do that pretty much using this file because it will have all the antibody responses such as HAI titers and not neutralizing antibody responses. There is also identifier called pathogen vaccine type. So you can also choose to do studies on different subsets of the vaccines. And as I said, we are going to filter the data in this tutorial to only include people under 50. And then group them based on the response. Any questions? Oh, so one thing I do want to mention is that for today's tutorial, you do not need to download the file from this website. In the tutorial that you have in this folder, we have already embedded the file in the tutorial, so we will directly import it in this Google Colab. So you do not need to download the file and upload those two RDS files that I was just talking about. So I'm just going to go a little bit forward and talk about the other modules. So I should also mention that the way you run Google Colab is that the code is written in these small pieces, which you can run independently. So if you can see that if you hover the mouse on this cell, you can see a play button, and basically that's how we will be running the code today, one by one. So in the first cell, right now, we are installing the packages. So unlike in R, when you are working with your own desktop version of R, you can already keep everything installed. And then you can directly call all the packages using library. Since this is running on Google Colab, we do not have all those packages installed. So we are installing the packages and then we are going to call them using library. And then this is the cell where we are going to import the two RDS files that I just mentioned. Those two RDS files will be read using read RDS and will be stored in ImmData and Asset objects. Okay, so once you have installed the packages, there should be a small green check mark over here, which shows that that cell ran successfully. So I hope it ran successfully for you as well. Now I'm just going to call all the packages and this runs pretty quickly. So again, I see that green check mark, which tells me that it worked. And now we are going to import the data sets. Okay, that ran on my website, my collab. I hope it worked for everyone. If you are not with me, please let me know. Okay, so now in this cell, we are going to now select the participants. So we are selecting participants younger than 50. And then assign them to the specific vaccine types. Okay, so the next step is data cleaning. So in this step, we retain only pre-vaccination samples, exclude studies with fewer than 10 samples to maintain the statistical power, remove the genes with any missing expression, and drop known outlier samples by the ID, participant ID. So now we want to run the machine learning algorithm, which is the random forest. And now we have to reshape the data so that our participant IDs are mapped to low and high responders, and gene expression data is available for random forest analysis. Okay, everything is running smoothly for me. So here we will define the high versus low responders. Again, as I said, you can see here MFCP50 is used in this analysis, and you can choose any other vaccine response metric for your own analysis. Okay, so let's run cross-validation. Any questions? Google Colab runs everything really smoothly, it looks like. I have never had any class where all audience or attendees have been so quiet. Okay, so here we are getting tenfold cross-validation prediction. We will calculate the ROC curve. So you can see here the true positive and false positives. Let's see, moving along. Okay, so now here is the information that we really wanted to look at, right? So we can look at the correct predictions, number of failed predictions for different vaccines. So we have influenza inactivated, influenza live, yellow fever, tuberculosis recombinant, and hepatitis vaccine. These are all the parameters for the random forest. We see sensitivity and specificity. And then we can make a plot out of it and have AUC calculated for each vaccine type. So after this part, so here is basically what we have for machine learning and development and prediction of the model. You can change the model, of course. You don't have to use random forest. Try different methods if you want. As I said, you can change the vaccine response, of course, do the analysis on other population, other parts of the cohort for specific vaccine, specific age type, develop covariate models using different covariates such as race, ethnicity. So there is a lot you can ask using this data set. And then after this part, we basically have some visualizations to think about, okay, what is important, right? Now we have random forest, and as biologists, we are never happy with just having the accuracies. So we want to know what is underlying those accuracies. So here we have some visualization to explore that. So here we just have a PCA plot. Again, you can run that and see how it is distributed across high and low responders and across different vaccinations. And you can see that both PC1 and PC2 have very low variance explained. So in this case, you can naturally go to other principal components and explore what genes come up in other principal components. Here we can look at predicted probability distribution for high responders and low responders. So the high responders are in pink, the low responders are in blue. Here we have a confusion matrix that we can run. So on x-axis here we have actual numbers, and on y-axis we have predicted numbers. So you can see that 165 high responders were correctly predicted as high responders and 97 were correctly predicted as low responders. And then there were some errors in the prediction. Here are top most variable genes across the entire data set. So here we have top 20 genes that have high importance. So now we can ask questions specific to each vaccine also. Here I'm showing you top 20 most variable genes just for influenza inactivated vaccine. You can ask if your favorite gene is in it. I see a CXCL10. Now we can look at 20 important genes for influenza vaccine in predicting high response. These are basically similar analysis as I showed you earlier but only focusing on influenza vaccine. And drop curve is again not too bad. So yes, that's all actually I have. Are there any questions? So everyone was able to run the tutorial?
Hey, Julie, thank you for a great presentation. We don't see any questions in the chat right now, but do encourage folks to raise their hand if they've got some questions. Oh, and I see a yes in the chat, likely for being able to run the tutorial successfully.
Perfect. So if we do have some time, I can download this data resource to show you what is in the data resource, unless there are questions about the tutorial. So you do not need, as I understand, you do not need to have ImmPort account to get access to this data resource. You just need to accept and continue your user agreement here. And if you do download it, I might have to stop share and share another file. I just want to show you what we have in that file.
Yes, Julie, the Google collab is running smoothly. What you just mentioned before, right? Everything is,
Yeah.
yeah.
Whenever I have ran these tutorials, most of the time we spend in installing the packages, but I think it went smoothly today. Let me see, where is... Hmm. Sorry. Um,
Yeah, we can hang around for a little bit longer.
yeah.
If you have
So now
questions.
do
Yeah,
you
no,
see this folder?
yes, we do.
Yeah, so now this is the folder I downloaded from the import, and I just wanted to show you if you do want to develop your own code or use it in any way. These are the RDS file I showed you. And we also have the CSV file, which basically has the same data. And there are also markdown files that you can use for developing your analysis. And then this is the README file. And the README file has all the information about all the columns, mostly in the metadata, where you really need a lot of information, because the other expression underscore data, or the ISET file, basically has gene expression data, but metadata has a lot of information by which you can actually develop your own classifier. So yeah, I wanted to just show that to you so that you can, if you want to develop your own analysis, this is useful. So do we need to, I think in the chat, I see question, please share the link. Do you want the link for data resource? Okay, yes, please. I don't have ability to send message to everyone, so someone else would need to.
Yeah, we will be happy to send it around. Okay.
Also, if you're looking for the link to the Google Colab notebook that Julie went through today, that was available in the participants, the materials for participants workshop portal. So depending on which link you're looking for,
I think they are looking for the resource page, import resource page.
Okay, great. Yep, Sanchita just shared that link as well.
Yeah.
Please let us know if that was not the link you're looking for, and we'd be happy to provide any further links.
And Julie, you want to show the tutorial link? That
Okay.
might be also helpful from.
Yes, just a second. Okay, so if you go to this folder, this is the folder that was shared with everyone. And you can go to the guided AI tutorial, and this is the link, Colab link, that you can open using OpenWidth in Colab for your analysis. And the data is also available in this folder, so the two RDS files. So now that we do have some time, I can also show you that here, the way we have, so here the data is directly coming from ImmPort website. So if you want to run it for your own analysis, this link, I'm not sure if will stay active. So you can actually use this sidebar, which has the folder icon, and over here you can see there is upload. So you can upload the two data sets that I was just talking about using basically browser. So it does take, if you go this route, it takes few minutes to upload the data, since the gene expression data is huge. So make sure you're patient while the data is getting uploaded. So you would need to upload the data and then change the link here. So you can basically just delete this part and only keep the file name, and this will directly read your data from that current Colab version. But if you now close the Colab, the data upload is going to time out. So Yeah, just so that you know, every time you run this Colab, you would need to upload the data. There are a couple of other ways to access the data as well through Google Drive um, in this for the Colab. Anything else? Any other questions?
Thank you, Julie. This is a great presentation and hands-on tutorial that you presented. I know people might want to wrap up a little early. And if you have any questions, just let us know. I will take just few minutes. Julie, let's watch out for any other questions.
Sounds
I'll
good.
just
Yeah.
yeah, and I'll just take few minutes to before we wrap up some information that I would like to provide. Share this. Can you see my slides?
Not yet, Sanchita.
Not yet. How about now?
Yep, we can see them now.
Okay, awesome. Right. So yes, I'll just spend a few minutes giving you information about the activities that we do within ImmPort, community engagements. So if you want to stay connected with ImmPort, we do organize webinars on a quarterly basis. Oh, so I think I was on the wrong page. The webinar, so we also publish the webinars on our web page. So here is this QR code if you want to go back and see the past webinars. And then also we send out the announcements for the upcoming webinars, and these are in YouTube format. You can download them, and also we make these slides available. And we invite researchers, those who have used data or data submitters. And so there's a different kind, all kinds of topics within ImmPort. We talk during the webinars. So please feel free to join when you can. And here's the QR code. And then if you want to connect with us, that you have data or you want to access the data, you have questions on data submission, please feel free. We have ImmPort office hours first Thursday of every month. So discuss your questions directly with the ImmPort team. By now you have maybe you're now familiar with these faces, so you will connect with them. And all user levels are welcome. Let's say that you don't have the data, but you want to get started. You can come to these office hours with your questions. Here is the QR code for the events page, office hours, so you can sign up. Newsletters, so we post newsletter on a monthly basis where we highlight on the new data that is getting released through ImmPort, resources, features, and events. So if you want to stay connected, please feel free to register, and you will get these newsletters in your inbox every month. We are also on the social media. Here are the links, and you can also stay connected to us using the social media. With that, I'll just come to end, and I would like to acknowledge and I would like to sincerely thank everyone who contributed this workshop. So most of the people that you have seen are, the names are here, and not only just contributing to this workshop, but to the ImmPort effort more broadly. And this truly has been a collaborative effort across Date leadership. Dr. Steve Kleinstein, who is the program officer, he's the principal investigator for ImmPort, Paraton, ICF, and other collaborators like University of Rochester. We have fundings from ODSS, ODSET, OTSAT, and last but not least, data providers, research participants, data consumers. We sincerely thank them. Without that, ImmPort won't exist. And thank you for your attention. Thank you so much for participating in our workshop. We truly appreciate your time, energy, and thoughtful contributions. And the questions and ideas and engagements helped make this session meaningful and productive. And we hope you found the workshop valuable and insightful. If you have any feedback for us, you can send us through help desk, or if you're looking for AI-ready data sets, go to the resource page, or you have any questions about how to submit the data, we have an extensive documentation, so go to the documentation page, and we have chatbots to help you navigating through the websites and so on. We have publications of data reuse. And if you have data in hand and how to submit the data, what templates can be used. And we have also data management sharing templates that you can take advantage of. Once again, thank you for being part of it. And we look forward to staying connected and seeing you at future events. Thank you.